Dear colleagues, it's my pleasure to greet you at the session Autoimmunity and Autoinflammation. I think it's very telling that in the past several years, more and more ESID meetings have talks and sessions on autoimmunity and autoinflammation. And I think that is because we now understand that these topics are not separate issues. They are part of immune continuum, if you will, which describes influence of monogenic inborn errors of immunity, polygenic predisposing factors, environmental factors, and probably other things that we don't even know about, influence on the multiple very different phenotypes of our patients. And I think it's very important also to blend the borders between rheumatology and immunology in order to study the pathogenesis of autoimmunity and autoinflammation in various settings. And I think this current session will be one more step towards this goal. With that, I will pass my virtual microphone to our first speaker, Dr. Husebye, who will be talking about the autoimmune regulator and its role in autoimmune diseases. Please.
Good morning, ladies and gentlemen. First of all, I want to thank the organizer for inviting me to speak on the role of autoimmune regulator in rare and common autoimmune diseases. My name is Husebye. I'm a professor at the University of Bergen and also affiliated with Karolinska Institute. These are my disclosures. And I want to start with a case report of autoimmune polyendocrine syndrome type 1, because study of this disease led to the identification of the autoimmune regulator gene. So this case was a young boy who at the age of 1 developed chronic oral candidiasis. A few years later, he was admitted in a critical condition with acute adrenal insufficiency and also hypoparathyroidism. He was then followed by his pediatrician and later presented with enamel hypoplasia, alopecia, nail dystrophy, and a severe malabsorption. Having both adrenal insufficiency and hypoparathyroidism, we could diagnose him with APS1 on clinical grounds. So the candidiasis is severe, but does only affect the mucous membrane, skins, nails, not invasive candida infections. Enamel hypoplasia is very prominent and affects most of the patients. Alopecia is usually severe and total, and many have this nail dystrophy. APS1 or APOSET, autoimmune polyendocrineopathy, candidiasis, ectodermal dystrophy, is one of almost 40 diseases, part of the Finnish disease heritage. And Finnish geneticists and clinicians together identified the gene behind this disease in 1997 and called it AIR, autoimmune regulator. And studies since then has shown that AIR is crucial for central immunological tolerance. So AIR has a particularly high expression in the thymus, in the medulla, and in a cell type called medullary thymic epithelial cells. And what AIR does in these cells, it turns on the transcription of tissue-specific proteins or antigens, proteins that normally are not found in the thymus, like insulin. These proteins are displayed in the context of MHC for developing T cells. And if there's an autoreactive T cell passing the MTEC, it will either go into apoptosis, and some of the autoreactive T cells will also be transformed into FOXP3 positive T helper cells. If ER is not working, these tissue-specific antigens are not expressed, and the autoreactive T cells will escape into the periphery, where they can give rise to autoimmunity. And the T helper cells will not be developed normally. So this gives rise to autoimmunity later. So mutations in ER give rise to APS1. I mentioned the main components, Addison's, hypopara, and chronic mucocutaneous candidiasis, but they can have a host of other autoimmune diseases affecting almost any organs in the body. They can have hepatitis, exocrine pancreatitis, insulinitis. Women affected very early and can go into menopause even in their teenage years or early 20s. A lot of patients have animal hypoplasia, alopecia, vitiligo, and so on, up to 20 different manifestations. So the disease has an autosomal recessive inheritance, but sometimes may be dominant. I'll come back to that. They also develop a lot of organ-specific autoantibodies, but also antibodies against interferons. So we have had a large interest in this disease and try to identify and study all the patients in Norway. And this is from our latest overview of the Norwegian cohort, 34 families. You see here from the age of onset of the first component that there's a huge variation. So what we see is a very diverse phenotypic presentation of this disease. They have an increased mortality. They can die of adrenal crisis, cancer linked to the chronic candidiasis, squamous carcinoma of the oral mucosa or the esophagus, but some also die of autoimmune components, hepatitis and pneumonitis. We identified ER mutations in most of our patients, and all our patients have at least one organ-specific autoantibody, and most of them have these interferon antibodies against type 1 interferons, especially alpha and omega. This is some more details on the disease components of our patients. As we have used now interferon omega as a screening tool to identify patients, we see more diversity. Only 40% have all the three main manifestations as the patient I mentioned had. So we see more patients with other phenotypes, milder phenotypes. The gonadal failure, ovarian failure, is very frequent in women. And we have this animal hypoplasia that can give a clinical clue to the diagnosis. As you see here, where the dentine of the teeth is exposed. Our patient illustrated what is quite common and traditional. Candidiasis comes first, followed by hypoparathyroidism and Addison's disease. Other endocrinopathies develop later in the course of the disease. The ER mutations, now about at least 130 different mutations are known. They are distributed through the whole gene, altogether 14 exomes. This shows the different domains. And the main mutation in Norway is the 13 base pair deletion. It's also very common in the British Isles and North America. But the most common one in the world is the so-called Finnish major mutation. It's a stop codon in exon 6, but it could also have been called the Russian major or the Polish major. It's very common in Eastern Europe. But usually we use the interferon omega antibodies to screen the patients, and you see here that this assay differentiates APS1 patients, this is Russian and Norwegian patients, from non-APS1 or blood donors very well. If they're positive, we sequence the AIR gene. Interesting thing about these interferon omega antibodies, they are sometimes seen in other diseases like myasthenia gravis, especially if they have a thymoma. But last week in Science, Paul Bastard in Casanova's group published that patients with life-threatening COVID-19 infections have interferon omega and alpha-2 autoantibodies, just like APS1 patients. So they found 10% of life-threatening COVID-19 patients had such antibodies, while those with asymptomatic mild COVID-19 did not, as healthy controls. The frequency in the healthy controls was about 0.3%. So these antibodies were present before the COVID-19, but they could bind the interferons and thereby making the route for the virus to infect more easily and the result a more severe infection. A few years ago, we stumbled over this patient with APS1. This index patient developed chronic mucocutaneous candidiasis and Addison's disease at the age of 25, so quite a late start for APS1. He also had pernicious anemia and typical antibodies, including interferon antibodies, and we sequenced the AIR gene, but found in this case only one mutation, P311Y. What intrigued us was that he had children with APS1-like phenotypes. So with his first wife, the daughter developed hypoparathyroidism and ovarian failure in her teenage years. With his second wife, he had 4 children. One had alopecia, one had no clinical phenotype, but APS1-linked autoantibodies, and the third, again, had hypoparathyroidism and ovarian failure. And these 3 children and this daughter all had the same mutation in the AIR gene, but only one mutation. And in this family, a dominant pattern of inheritance. So we asked, could there be dominant mutation, and what could be the explanation? In addition, this daughter got a son who at the age of 1 developed interferon omega antibodies, but still he has now at the age of 2 no phenotype. When we looked at the literature, we found this mutation had been reported in Finland in 2 sisters, also with the Finnish mutation. These developed a full triad of APS1 with CMC, Addison, hypoparathyroidism in childhood. When we investigated the family, it turned out that one of the patients had a son who developed severe pernicious anemia in childhood and severe vitiligo. He only had this C311Y, the same mutation as our family had. And also the grandmother had this mutation and developed severe pernicious anemia. So in this particular family, you see classical APS1 childhood onset with 2 mutations, with 1 mutation, a milder phenotype. So C311 is located in a critical place in the protein, so it's in the PHD1 domain, excuse me, and a cysteine that binds or coordinates the zinc in this PHD zinc finger is substituted with a tyrosine, and you can envision that the tyrosine will block the binding of the zinc, and if you simulate this change, the whole globular domain will disintegrate and become unstructured and probably not functional. So we tried now, set out and had the hypothesis, could it be a dominant negative effect? by this mutation. So this is a bit busy slide, but if you focus on this panel, here we tested human thymic 46 cells. We transfected the cells with the Finnish major mutation, so non-functional, truncated protein, and ask what happens with ear-regulated genes when we transfect with the Finnish mutation. As expected, no ear-dependent transcription takes place. But if you co-transfect the Finnish mutation with the wild type, you can rescue ear-regulated transcription. Likewise, with this variant in the CAR domain of the ear, we see the same rescue when we titrate with increasing amount of wild type. On the contrary, looking at our mutation from our family, with the mutated ear alone, no ear-dependent transcription. But when we try and rescue with the wild type, we still see no ear-dependent transcription. So it seems like a dominant negative effect. We also tested this mutation in the SAN domain, and found in a family with also dominant inheritance pattern. And again, we see no rescue by the wild type. So it seems that these two, C311Y and the G228W, have a dominant mode of action, while the Finnish major and this CAR domain variant show a kind of recessive mode of action. We then looked at the literature, and strikingly, many of the patients with PHD1 mutations, in these patients, the investigators found only one mutation, not two. So it was explained maybe there was another mutation in a regulatory domain or a promoter or something but it wasn't considered that it could be dominant. But when we tested a lot of these variants here in red, we saw the same dominant pattern when tested in our 4D6 cell system. And if you compile the families we characterized and looked at the phenotype and also autoantibodies, we found that interferon autoantibodies was less frequent, the main manifestations were less frequent, but many of the patients displayed vitiligo and pernicious anemia. And some carriers of these variants had no phenotype, so the penetrance seemed to be reduced. So this can be transformed into kind of a model. You have normal ear, it's functioning, you don't have autoimmunity, you are healthy. If you have one mutation in the PHD1 domain, a dominant one, you get autosomal dominant APS1, mild phenotype, often vitiligo, pernicious anemia, while if you have two mutations in the ear gene, you get the autosomal recessive classical APS1 with early start in childhood and the main manifestations. This was shown now in the cell system, and it's not unreasonable to think that it's a dominant negative effect because ear is operating as a dimer or probably a tetramer. So if you have a mutated protein together with a wild type, this change in the structure of the mutated protein could inhibit the wild type's action. But we then set out together with Jakob Abramson at the Weizmann Institute and Yael Goldfarb did these experiments, tried to replicate these findings in a mouse model. So we made a number of mutations, two recessives, one in the CAR domain. This is in the same codon as our family's mutation. So C311 in human is the same as C313 in the mouse. This is a stop codon, supposed to be recessive. This is our dominant mutation in the mouse. So Yael isolated MTEX and asked how these mutations affected ear-dependent transcription by looking at the whole transcriptome. So if you look at this panel first, where you compare a heterozygous mutation with an ER knockout. So this is the log2 fold change wild type over het plotted against log2 fold change wild type over ER knockout. And you see in the ER knockout, ER-dependent genes in red are not transcribed, but they are in the heterozygous mutation. You see the same for both of these. But if you have the homozygous mutation, both alleles with one of these variants, you see it behaves like the ER knockout with inhibited ER-dependent transcription. On the other hand, if you do the same type of experiment with this dominant mutation, you see another pattern where in the heterozygous condition, you see it behaves like the ER knockout with inhibited ER-dependent transcription. So it looks like it has a dominant mode of action. Looking at these mice, they develop a wasting disease in the heterozygous condition. They develop autoimmune infiltrates in a number of organs that are affected in the ER knockout model, and they have a wasting disease and decreased survival in this model. So to sum up this part of the presentation, ER causes autoimmune polyendocrine syndrome type 1, but in some cases with a dominant mutation, and these variants have a frequency in populations of maybe 1 in 1,000. You can see a milder phenotype that masquerades as autoimmune polyendocrine syndrome type 2. So what is type 2? Well, it's the combination of Addison's disease with autoimmune thyroid disease and or type 1 diabetes. So Addison's disease is most often seen in this context with these diseases, and actually the majority of Addison's patients have, in addition to Addison's, one of these two other diseases. So we asked, is ER implicated in other autoimmune diseases, specifically in autoimmune Addison's disease, since APS1 and APS2 have this particular component in common. So we have just performed a GWAS of the Norwegian and Swedish Addison registries, compiling two national registries with very carefully phenotyped 21-hydroxylase positive Addison patients. So 21-hydroxylase autoantibodies is a biomarker for autoimmune Addison's disease, equivalent to what GAD65 is for type 1 diabetes. We also picked controls from different parts of Norway and Sweden to reflect the population throughout the countries, and did a traditional GWAS asking if there are variants, SNPs in the genome that segregates in cases and not in controls. And these are still unpublished data, so I won't go into too much detail, but as expected, we found a very strong association to HLA. This was known. We had a p-value of 10 to the minus 179, so a very strong association. We also replicated earlier findings showing association to Beck2, to CTLA4, to PTBN22, but we also found some new association. And what was particularly intriguing was the association to ER, a very strong association to ER. And this is a coding variant. It changes amino acid that could be critical for one of the PHD1 the PhD domains. And we think that this subtle change in the structure probably renders these patients more susceptible for autoimmune Addison's disease. So ER can cause rare disease, APS1. You have in between these dominant mutations giving rise to a milder non-classical APS1, often segregating in families with several autoimmune features, but also ER is clearly linked to the common type of Addison's disease, and we saw no difference in association to this ER variant if patients had isolated Addison's disease or APS2. So to summarize, mutations in ER cause APS1. The main manifestations are Addison's disease, hypoparathyroidism, chronic mucocutaneous candidiasis. There's a large phenotypic variation. If you only use the classical main manifestation to screen for patients, you will miss many. So interferon alpha and omega autoantibodies is a very strong tool to screen patients, and positive patients should then be ER sequenced. Usually you find two mutations, a recessive mode of inheritance, but some of the variants are dominant and shows a dominant negative effect. They might start in adulthood and have a milder phenotype. ER is crucial for central immunological tolerance, and changes here seems to predispose patients also for the common Addison's disease, the kind of regular autoimmune Addison's disease. And intriguingly, these interferon antibodies seems to be important also for the susceptibility for severe COVID-19 infections. Finally, I want to acknowledge the KG Jepsen Center for Autoimmune Disease, a major funding source for us, and all our collaborators, especially in Stockholm, led by Ole Kemp at the Weizmann Institute, led by Jakob Abrahamsson, and other collaborators and funding sources. Thank you very much for your attention.
Hi, I'd like to thank the organizers for selecting my abstract for a talk. My name is David Beck, and I'm from the NIH, and I'll be presenting today about a new genetic disease that we've identified in the lab. So I work in Dr. Dan Kastner's group, and we're interested in identifying the genetic causes of autoinflammatory diseases. And typically we do this by analyzing families, performing whole exome sequencing, and looking for variants that can explain phenotypes. But more recently, we've been taking an approach where we look across our whole cohort, independent of the specific family or symptoms, and see if there's any shared variation that can explain potential phenotypes. So I performed this genotype-first approach, looking at a pathway, protein ubiquitination, and I picked this pathway because we've identified multiple different autoinflammatory diseases that are caused by mutations in genes that regulate ubiquitination. I looked across the exomes in our cohort and the NIH undiagnosed disease cohort, specifically at genes that are intolerant to haploinsufficiency, novel mutations not present in NOMAD, and mutations shared across multiple cases. And we identified 3 middle-aged men, all with inflammatory disease, all with mutations at the same codon in UBA1. So what was initially confusing about this is that UBA1 is an X chromosomal gene, and these men had seemingly heterozygous mutations, but after some genetics detective work, we were able to identify that this mutation is somatic and restricted to the blood. And not only is it restricted to the blood, but it's restricted to specific lineages. So the wild type, these are Sanger chromatograms, the wild type alleles in red and the mutant alleles in blue. As you can see in the earliest precursor cells, there's a mixture of mutant and wild type cells. But as we look at the more differentiated cells in the periphery, neutrophils and monocytes are predominantly mutant, whereas T and B cells are predominantly wild type. And what's interesting is that the T and B cells are decreased in these patients, suggesting that there may be some selective disadvantage for the mutation in those lineages. We can quantify this effect, and here I'm just showing that the fibroblasts and T and B cells have no detectable mutation, whereas the myeloid cells and precursor cells from the bone marrow all have the mutant at high VAF. And not only do these patients have similar inflammatory phenotypes, but they also have these unique features in their bone marrow where their erythroid and myeloid precursor cells all had cytoplasmic vacuoles present. This is highly characteristic of this disease and shared across all individuals. Using the clinical information from the first 3 patients along with the vacuoles, we were able to identify 25 cases, and this number keeps going up. All of them men, all of them with late onset disease. It's a severe disease with 40% of the patients deceased. And it's actually very treatment refractory, and patients only respond to high dose glucocorticoids. The clinical symptoms include fever, skin, lung, cartilage inflammation, and then hematologic manifestations including clots, macrocytic anemia, and the bone marrow vacuoles. Patients actually meet criteria for multiple different diagnoses. So some of them rheumatologic, relapsing polychondritis, Sweet syndrome, some of them hematologic malignancies, myelodysplastic syndrome, and multiple myeloma. And even the patients who have hematologic malignancies, those malignancies are preceded by an inflammatory disease. So this mutation in all 25 individuals leads to different diseases. We've named this disorder VEXUS for vacuoles, E1 ubiquitin activating enzyme, X-linked autoinflammatory somatic syndrome. And this is just showing the top row, the pulmonary, the second row, the skin inflammation, the cartilage inflammation, the third row, and then finally the hematologic manifestations with a PET scan showing inflammation in the bone marrow. And we know in these patients that the inflammation is driven by the mutant myeloid cells. So here we're just looking at electron microscopy in VEXUS patient neutrophils. You can see that the cells are very disordered and they're actually undergoing necrosis with all the cellular debris outside. And when we did sorted cell RNA-seq, what we saw is that in the whole blood and sorted neutrophils and monocytes, there was upregulation of multiple different inflammatory pathways, whereas in the non-mutant lymphocytes, these pathways weren't upregulated. So this is showing that these mutant cells are driving the inflammation. So switching gears, ubiquitination actually occurs through 3 distinct steps. The first is E1 activation. So there are 2 enzymes, followed by 40 E2s that work with 800 substrate-specific E3s. And these work together to ubiquitinate over 50% of the proteins in the cell at any one time. So UBA1 is responsible for over 99% of the ubiquitination in the cell. It's one of the E1s. And UBA1 has 2 translationally regulated isoforms. The first is initiated from methionine 1, and this is a nuclear isoform, UBA1A, and the second is initiated from methionine 41. This is a cytoplasmic isoform called UBA1B. And all of our patients have mutations at the initiating codon for UBA1B, methionine 41. So we hypothesized that this disease would be caused by loss of the cytoplasmic UBA1. But unfortunately, when we looked by immunofluorescence, this wasn't the case. So we next looked at the protein levels in patient cells. And as I mentioned before, T cells are wild type in patients. And when we looked at the isoforms for UBA1A and B in patients versus controls, there were similar levels, and so were the levels of polyubiquitination. However, when we looked at mutant monocytes from patients, you can see that there's a loss of the UBA1B band and emergence of this faster migrating UBA1C band. And along the lines that this mutation is a loss of function, we can see there's a decrease of polyubiquitination and an increase in the substrate, free ubiquitin. So given that this methionine is a start codon, we hypothesize that the new isoform may be an alternative translation product downstream. And we can recapitulate this faster migrating band in vitro. So when we express methionine 41 mutations, these lead to a faster migrating band. And when we mutate the next coding methionine 67, you can see that this band is abrogated. So this is demonstrating that the new faster migrating band is actually a novel isoform initiated from methionine 67. And we can look at the catalytic activity of this UBA1C, and it's actually impaired compared to wild type. So in terms of thioester bond formation. So together, this is showing that VEX's mutations lead to loss of the catalytically active cytoplasmic UBA1B and emergence of the catalytically impaired UBA1C. And we can model the inflammatory effects in zebrafish. So this is showing transcript expression of different inflammatory pathways. And in the complete knockout in blue or the cytoplasmic knockout in green, you can see there's activation of these, there's increase in these transcripts, whereas in the nuclear form, when we knock out UBA1A, they aren't increased. So this phenocopies the patients. So together I've shown that this is a mosaic disease caused by mutations in UBA1 that's restricted to the myeloid cells and leads to also a decrease in lymphoid cells and an alteration in the isoform. So there's loss of the normally active UBA1B in the cytoplasm and emergence of the catalytically impaired UBA1C. So just to summarize, I've shown today that the genotype first strategy can identify diseases that weren't thought to be linked together that had similar genetic etiologies. I've also identified that somatic variants on the X chromosome are an underrecognized cause of disease. And finally, that somatic mutations may account for a significant fraction of adult onset inflammatory disease. And with that, I'd like to thank my mentor, Dr. Kastner, Ivona Oksentievich, and the tremendous help I've had with this project. And then for any questions you have or potential referrals, here's my email information. And thank you for your attention.
So good morning, everyone. I am Marie-Louise Frémond. I am a pediatrician working as a postdoctoral fellow in the Crow Laboratory in Paris. And it's a pleasure to share with you our work on COPA syndrome and new insight in disease pathogenesis. I've got nothing to disclose, and I kindly remind the audience that questions can be submitted during the talk in the top right corner of the screen, and that this question will be addressed during the online live question and answer section at the end of the session. So heterozygous mutation in COPA were described in 2015 to cause an autoinflammatory disorder demonstrating joint, lung, and renal involvement. High titers of autoantibodies, together with evidence of ER stress and the priming of the Th17 response, were also described. To date, less than 60 patients were reported in the literature, and there is a remarkably high clinical non-penetrance. However, the precise pathophysiology of this disorder is not properly understood. COPA is a subunit of a COP1 complex that is described to be involved in the trafficking of cargo proteins between the GOLGI and the ER. And actually, there is a striking clinical overlap between COPA syndrome and SAVI, a well-defined monogenic type 1 interferonopathy, in particular regarding the joint and lung phenotype. More recently, Volpi and colleagues described an increased expression of interferon-stimulated gene, ISG, in the blood circulating cells of several COPA patients. So we asked the question whether COPA syndrome could be a new type 1 interferonopathy. To do so, we recruited 9 patients, all carrying a mutation either reported or located at the same amino acid substitution position. And of interest, all of these mutations shown here in red are located in the VW40 domain of the protein that is involved in the recognition of the cargo protein. And of further interest, 2 more recently identified variations shown in yellow and orange are close to but distinct to this hotspot. The phenotype in our cohort were as previously described. Considering the possible shared pathophysiology between SAVI and COPA syndrome, we compared lung biopsy tissue from a SAVI patient and a patient of our cohort, and observed similar features such as lymphoid follicles filled with CD20 plus B cells, macrophagic alveolitis, and emphysema. Of course, one shouldn't consider this lesion as specific. We then aim to assess the interferon pathway activation in COPA patients, and we record high levels of interferon alpha protein per se using a CIMOA ELISA, and high interferon score in COPA symptomatic patients, whereas COPA asymptomatic carriers displayed normal or low values. To decipher the interferon pathway activation seen in patients, we use a cellular model of HEX293T cells that lack STING expression. And we observe that IL-3 that is needed to induce the interferon production was phosphorylated only in cell co-transfected with STING wild type and any of the COPA mutant plasmid that we use. And this was not the case in cell transfected with COPA and STING wild type or in cells transfected with a COPA mutant plasmid and empty vector. Similarly, increased expression of IgG interferon beta was only seen in the presence of STING wild type and the COPA mutant plasmid. Then we wanted to assess the mutation mechanism, and we sliced COPA in THP1 cell monocyte cell lines that are competent for STING signaling to interferon. Doing so, we observed that phosphorylation of IL-3 and expression of IgG and interferon beta was induced in cell knockdown for COPA, and this was abrogated in STING KO cells. This data suggests that COPA mutations act in a dominant negative manner, and this confirmed the STING dependency seen in the previous experiment. STING-dependent interferon induction involves intracellular trafficking, and it is known that gain-of-function mutation in STING leads to constitutive activation and translocation of STING to the GOLGI. And of interest then, in HEC293FT, we observed that STING wild type had an increased localization to the GOLGI when the cells were co-transfected with a COPA mutant, equivalent to what observed with the STING gain-of-function mutant V145M. What about treatment? Giving our data, 3 patients of the cohort were trialed with ruxolitinib, a JAK1 and 2 inhibitor, including 1 patient with a severe lung phenotype. We observed a clinical improvement, but progression to lung fibrosis on chest CT scan, and more recently, a clinical relapse, requiring the switch to baricitinib, another JAK1 and 2 inhibitor. This warrants further combined or alternative treatment for patients. In conclusion, our data characterize COPA syndrome as a new monogenic type 1 interferonopathy. And we speculate that COPA has a role in the retrograde trafficking of STING from the GOLGI to the ER. This also highlights the ER-GOLGI axis as a new focus for research, and the potential for drugs targeting this axis in STING-dependent autoinflammation. With this, I would like to thank you for your attention. I would like to thank the patient and the families, all my lab, in particular Yannick Rowe, Alice Lepellet, and Maria Martin-Nicholas. Thank you for your attention.
Thank you very much for the invitation to present our work. I and my collaborators have nothing to declare. So NF-kappa B refers to a family of 5 transcriptional regulators largely maintained in a latent state by inhibitors of kappa B. Activation via either the canonical or non-canonical pathway leads to degradation of inhibitors and transient liberation of the transcriptional regulators. NF-kB2 encodes P100, which is both a member of the IKB family and also a precursor of P52. Now, human NFKB2 syndrome arises from heterozygous mutations that are clustered in the C-terminus of the P100 molecule and result in heterogeneous syndrome that includes various forms of organ-specific autoimmunity. We set out to elucidate the mechanism of autoimmunity in this syndrome. And we generated an NFKB2 allelic series by CRISPR-Cas9. These strains were complemented by the LIM1 strain, which had been generated more than a decade ago by ENU mutagenesis, a strain completely deficient in P100 and a strain deficient in AIR. Protein analysis of splenic lysates is illustrated in this representative Western blot, and the collective data are summarised in these diagrams. And what can be observed is that there is processing resistance of P100 to P52 in the D865G LIM1 and 868 indel strains, an overall reduction in P100 and P52 in the 868 frame shift, and no P100 or P52 in the Zander strain, the deficient strain. Most of the strains survived normally, but those heterozygous for D865G and for the LIM mutation had shortened lifespan, and this was more marked when we made the mice homozygous. The curtailed lifespan correlates well, or is inversely correlated at least, with the P100 to P52 ratio rather than the overall abundance of P52 or P100. Now, the shortened lifespan is T-cell dependent. When we rendered the LIM strain T-cell dependent by crossing to a TCR alpha deficient strain, the mice survived normally. Pathology is characterised by exochronopathy, polyexochronopathy, and it's noteworthy that the severity of pancreatitis and the absence of prostatitis and retinal inflammation distinguishes this pattern of end organ pathology from that observed with AIR mutants. We conducted a reciprocal bone marrow chimer experiment, and this revealed that mutations in the non-hemopoietic recipient component conferred the reduction in survival, as shown here, where LIM recipients of wild-type bone marrow had the poorest survival. By contrast, when we transferred in mature hemopoietic cells, there was no effect on survival, indicating that the defect arose when mutant cells arose in the mutant environment. So this prompted us to look at the thymic epithelium, and analysis of these histological sections reveals that there is a considerable reduction in the thymic medulla in strains carrying processing resistant versions of P100. And this is in contrast to what is observed in P100 deficient strains and indeed in AIR deficient strains. FACS analysis of single-cell suspensions from the thymus revealed a reduction in medullary thymic epithelial cells, thymic B cells, and thymic dendritic cells. And again, this phenotype is in contrast to that observed with either AIR deficiency or complete P100 deficiency. We looked into this intrathymic defect in greater detail using a strategy where we were able to track thymocytes during development. In the presence or absence of a BCL2 transgene that reduces apoptosis and therefore helps to reveal cells that would otherwise be destined for deletion. Wave 1 of intrathymic deletion is marked by registering a strong TCR signal and is characterized by expression of Helios but not CCR7. And the D865G mutation results in a reduction in this wave 1 of negative deletion. We observed a similar phenotype with the LIM1 strain. 2 days later in thymic ontogeny, cells destined for wave 2 of deletion or selection into the Treg compartment are marked by the presence of Helios and CCR7. And again, we observed a reduction in this population in those strains carrying a processing resistant version of P100. In humans with NFKB2 mutations, there's a reduction of regulatory T cells in the periphery, and indeed this is a more marked deficiency than is observed with AIR mutations, and this was recapitulated in the mouse strains. We see that in the P100 processing resistant strains, Treg deficiency, which is more severe than is observed with AIR or P100 deficiency. This also appears to be as a result of defects extrinsic to the hemopoietic compartment, construction of bone marrow chimeras with wild-type bone marrow into either wild-type or mutant recipients revealed that recipient genotype determined Treg deficiency. Next, we had a look in the periphery to see if this defect in thymic selection translated into a signature of autoreactivity in the T cell compartment. And we did this by enumerating T cells using a tetramer of IAB and PDIA2, a pancreas-specific self-antigen. And this reveals that indeed pancreas-specific autoreactive T cells are expanded, at least in the NFKB2 LIM heterozygotes. Finally, we performed deep sequencing of the TCR alpha and beta genes in various peripheral T cell compartments and thymic T cell compartments. This summary slide shows what's called a hydrophobicity index, which reflects the presence of autoreactive, reflects the particular motifs at positions 6 and 7 of CDR3 of TCR beta that confer autoreactivity. We found enrichment for this motif in conventional CD8 cells in the thymus and in the periphery, and also in the CD4 compartment in the periphery, in various strains with processing resistant versions of P100, but again not in AIR mutants. And remarkably, when we performed TCR beta sequencing in patients with NFKB2 mutations, we saw a similar enrichment for this autoreactive motif. So we've concluded that autoimmunity arises from mutations in NFKB2 that affect P100 processing. Pathology arises as a result of defects in thymic selection. Both pathology and functional defects are distinct from those observed with either P100 deletion or AIR deficiency. And collectively, these findings show that T cell tolerance is exquisitely sensitive to the I-kappa-B function of P100. I'd like to acknowledge my collaborator and co-lead on this project, Steve Daly. Rashika Wirasena and Ainsley Davies led the experimental work, and I'd like to thank and acknowledge our many collaborators on this project.
Good morning, everybody. I'm Lorenzo Lodi. I'm a pediatric resident at Mayer Children's Hospital, University of Florence, and I'm chief student at the Laboratory of Neurogenetics and Neuroinflammation directed by Yannick Crow at Imogen Institute in Paris, where I conducted this study about type 1 interferonopathies and interferon alpha levels in the cerebrospinal fluid. The project was supervised by Marie-Louise Frémond. I've got nothing to disclose, and I kindly remember to the audience that questions can be submitted during the talk via the question and answer function in the top right corner of the screen, and this question will be addressed in the live question and answer section at the end of the session. The upregulation of type 1 interferon signaling is common to all type 1 interferonopathies, but clinical disease expression varies significantly across this set of diseases, as you can see from the prominent systemic involvement of sting-associated vasculopathy of infancy, represented here by these overt skin features, to the prominent neurological one of Aicardi-Goutier syndrome, with intracranial calcification serving as an important indicator of enhanced interferon activity. In fact, enhanced interferon activity in the brain of AGS patient has already been demonstrated indirectly by Pierre Lebon team through antiviral-based cytopathic effect assay, but the direct quantification of these cytokines has long proven challenging. So in the present study, we first aim to directly quantify interferon alpha protein in the cerebrospinal fluid of monogenic and polyfactorial interferon-related disorders, and to compare this level with those of autoinflammatory disorders that were not interferon-related and with controls. Secondly, we aim to compare interferon alpha protein in the cerebrospinal fluid of interferon-related disorders with serum plasma interferon alpha concentration. To pursue our aims, we use a single molecule array ultrasensitive digital ELISA technology associated with a pan-alpha antibody obtained from APESID patient. And then we retrospectively recorded all patients referred to our laboratory for the evaluation of interferon alpha protein concentration in the cerebrospinal fluid between December 2014 and to December 2019. A concomitant or a closest measurement of interferon alpha in serum plasma was recorded when available. Patients included were subsequently assigned to one of the 5 disease groups presented in this slide. So as you can see, AGS and SEBI patient had to present with a causative mutation in one of the known genes. Neurolupus had to fulfill EULAR 2019 criteria, while for putative type 1 interferonopathies, clinical and laboratory criteria were specifically designed to demonstrate a constitutive activation of type 1 interferon pathway. All patients in the putative type 1 interferonopathies group underwent genetics testing, but without the identification of a known causative mutation. Similarly, design criteria were used for autoinflammatory disorders that were not interferon-related, for which the clinical response to anti-interleukin-1 treatment was considered corroborative. Patients with non-inflammatory hydrocephalus were included as controls. So for AIM-1, we recruited 42 patients belonging to the different disease groups cited before, and to avoid an analysis bias, only one sample per patient was analyzed. We observed that the median level of interferon alpha in the cerebrospinal fluid was elevated in AGS, SAVI, presumed type 1 interferonopathies, and neurolupus when compared to autoinflammatory disorders that were not interferon-related and to controls. And as you can see, the highest interferon alpha levels were recorded in AGS. Despite the small number of patients, we also observed that median interferon alpha level in the cerebrospinal fluid of AGS were similar across the different genotypes, as shown in figure 1. And then the youngest AGS patient had the highest interferon alpha concentration in the cerebrospinal fluid, as you can see from figure 2. And this was consistent with what previously reported with interferon activity. For what concerns our second aim, we consider all matched samples available per patient, including serial results from the same patient, and observe that in patients with AGS, interferon alpha level in the cerebrospinal fluid were higher than in serum plasma matched sample. And by contrast, in SAVI, presumed type 1 interferonopathies, and neurolupus, interferon alpha levels in the cerebrospinal fluid were lower than serum plasma matched sample. Of interest, always with regard to the comparison between cerebrospinal fluid and serum plasma, and considering only one matched sample per AGS patient collected under no treatment, we observed that only 2 AGS patients presented higher interferon alpha in serum plasma than in the cerebrospinal fluid, and intriguingly, both of them presented a lupus-like clinical phenotype. So, in conclusion, we show that interferon alpha is present in the cerebrospinal fluid and serum of all interferon-related diseases studied here. We also show that the primary site of interferon production in AGS and SAVI seems to be respectively the central nervous system and the periphery, and this may reflect tissue specificity or biological redundancy of the mutated gene and or in the generation of self-nucleic acids presumed to trigger the observed interferon response. Thank you for the attention. I want to thank, first of all patients and family, Yannick Crow, who allowed me performing this study, Marie-Louise Frémond, who constantly supervised me during this work, all colleagues in Paris and Edinburgh, and all, obviously, all clinicians who participate to this study. Thank you.
Good morning, everyone. First of all, I would like to thank the organizers for the invitation and the opportunity to share our most recent data on autoimmunity in WHIM syndrome. A quick disclosure, the study was partly funded by X4 Pharmaceuticals. I will start with a short introduction on WHIM syndrome. WHIM syndrome is a rare autosomal dominant immunodeficiency caused by a gain-of-function mutation in the chemokine receptor CXCR4. This gain-of-function mutation causes receptor hyperactivation that leads to leukocyte retention in the bone marrow, resulting in severe chronic panleukopenia. WHIM is extremely rare. On a molecular level, this gain-of-function mutation in CXCR4 leads to a truncation of the C-terminus and to deficient receptor internalization. And this subsequently prolongs the interaction of CXCR4 with its ligands and receptor hyperactivation. And this hyperactivation results in an inability of leukocytes, especially neutrophils, to migrate from the bone marrow to the periphery. That leads to an accumulation of mature neutrophils and other lymphocytes and monocytes in the bone marrow. In the bone marrow, the neutrophils undergo apoptosis, decreasing the amount of neutrophils in the periphery and resulting in a hallmark of WHIM in myelocathexis seen in this image below here. So WHIM syndrome is characterized by a wide clinical spectrum of disease manifestation, including susceptibility to infection, neutropenia, leukopenia, hypergammaglobulinemia, HPV-associated infections, especially warts, and in some cases, congenital malformation and vascular abnormalities. In recent years, there has been evidence that CXCR4 signaling might be implicated in the development of autoimmunity. However, there is no data on autoimmunity manifestation in WHIM syndrome. We therefore recruited an international cohort of 65 patients with genetically confirmed WHIM syndrome originating from 27 centers all around the world. We identified 15 total cases of WHIM patients with prominent autoimmune and inflammatory manifestation. This is a prevalence of around 23%. Demographically, we observed a slight or a significant predominance of female patients compared to WHIM patients with autoimmunity. All WHIM patients were white non-Hispanics, and they were significantly older than those without autoimmunity, with a median age of 21 years compared to 9 years. So the most frequent autoimmunity complication was cytopenias, followed by endocrinopathies in 3 cases, 2 cases of type 1 diabetes and 1 case of autoimmune keratitis, skin manifestation, including 2 cases of psoriasis and 1 case of vitiligo, and 1 individual or 2 individual cases presenting with arthritis and hepatitis. In total, 60% of our patients were had more than one autoimmune complication, especially cytopenia, were more likely to present with additional autoimmune complication, whereas other autoimmune manifestation were more likely to occur isolated. On the next slide, we see that ITP, immune thrombocytopenia, was the most frequent autoimmune complication in 9 patients, followed by autoimmune hemolytic anemia with 6 cases and 1 case of autoimmune neutropenia. Evans syndrome was observed in 3 patients, and pancytopenia with autoimmune neutropenia in 1 case. On the right side, you can see the severity of cytopenias during flares. There was a median hemoglobin level of 5.5 and a median platelet count of 70,000 thrombocytes. Therapy, the majority of patients, in the majority of patients, first-line therapy was used, including immunoglobulins and steroids. These first-line therapies were not sufficient to control cytopenia and needed an therapy escalation to second-line therapy, including, in the majority of patients, rituximab, B-cell ablation, and thrombopoietin receptor antagonist. These second-line therapies improved outcome of cytopenia. Yeah. So, in conclusion, autoimmunity and inflammatory manifestation are frequent complication in WHIM syndrome with a prevalence of 23%. WHIM patients with autoimmunity are predominantly female and older than those without autoimmunity, and most patients present with more than 1 autoimmune complication. Autoimmune cytopenia, especially ITP and hemolytic anemia, are the most frequent manifestation of autoimmunity. And autoimmune cytopenias are refractory to first-line therapies and need second or third-line treatment for definitive management. And with that, I would like to thank a couple of people, especially my PI, Johan Walter, Sarah Cohn, Marissa Messing, Rachel for the help in the preparation of the manuscript, our collaborator, especially Anna Shcherbina. Thank you very much.
Hi, I'm Natalia Hammerschmidt from the CCI in Freiburg, and I will talk about AD2 and it being a lysosomal DNase regulating the type 1 interferon response. We have to disclose that the group of Max Warnke from Novartis was a collaborator in this project. So AD2, the enzyme, is in current consensus considered to be an extracellular adenosine deaminase. However, this is a matter of intense debate because of the low concentration of the substrate adenosine in the extracellular space. And that is not at all in the range of the Km value of AD2. Also, when comparing the deficiency of a known adenosine deaminase, ADA1, to ADA2, they are completely different. So ADA1 causes severe forms of SCID, as you all know. ADA2 deficiency, however, causes a broad range of symptoms, and that was first described as polyarthritis nodosa, and now is recognized for being an immune dysregulatory syndrome with inflammation, bone marrow dysfunctions, and immunodeficiency described. So the mechanistic link between this hyperinflammation and the ADA2 function are largely unknown, though. And to address this link, you need to consider that AD2, contrary to AD1, which is expressed ubiquitously, is preferentially expressed in monocytes and macrophages. So that is why we looked at the proteomes of monocytes of 6 patients with biallelic mutations in AD2, and the pathway analysis shows an interferon signaling upregulation. The network analysis links this upregulation to the innate response to viral infection. And when looking at the heat map more closely, a lot of the differentially expressed proteins are involved in the interferon signaling. Those are marked in red here. So now the question arises if this observed effect is caused cell intrinsically. And to answer that, we produced an AD2 deficient monocytic cell line, a THP1 cell line, with the CRISPR-Cas9 technique. And the knockout led to an upregulation of the interferon-stimulated genes, as you can see here, as well as the secretion of CXCL10 that was also upregulated. And importantly, this upregulated of the interferon signature could be completely reverted by the re-expression of AD2 by lentiviral transduction, which you can see when comparing the red to the gray bars. So after observing the cell intrinsical effect, we looked at the intracellular localization of AD2 in biopsies of human tonsils from healthy donors. And as you can see, AD2 is localized in the tingible body macrophages and vesicles that with immunofluorescence were shown to be LAMP1 positive lysosomes. And to further confirm this, we analyzed the glycan structures of AD2 and compared them to the known lysosomal proteins. And as you can see, they cluster. And so this shows AD2 is sorted to the lysosome via the mannose-6-phosphate dependent pathway. So because of this lysosomal localization and the interferon response upregulation, we hypothesized that AD2 is involved in nucleic acid sensing. And to reevaluate its function, we looked at its structure. And here in the superimposed AD1 and AD2 proteins, you can see that AD2 possesses a domain, the PRB domain, which in our modeling was predicted to bind to double-stranded DNA. And to test the hypothesized DNA's function, we incubated endogenous porcine and recombinant human AD2 with DNA at different pH and cell concentration. And the DNA was degraded with a pH optimum of 4.6 to 5, which is corresponding to the lysosomal pH, and at low cell concentrations. Further, we compared the functions of AD2 variants that cause a clinical phenotype and evaluated the adenosine deamination and the DNase activity. And as you can see in this left graph, they correlate strongly. And so this suggests they rely on similar structural features of the enzyme. So the data up to this point suggests that the defect in AD2 leads to defective degradation of double-stranded DNA, which would subsequently leave more ligands for the rest of the DNA sensing machinery, and also would explain the interferon signaling upregulation. So to test this model of pathogenesis, we stimulated our AD2 deficient monocytic cell line with agonists of the pattern recognition receptor, namely poly-IC, LPS, imicule mode, and interferon stimulatory DNA. And as you can see in the upper 3 graphs, the first 3 agonists didn't change the interferon response. The interferon stimulatory DNA, however, led to an upregulation of the interferon production upon stimulation. And this could be completely reverted by the overexpression of AD2 or the genetic co-deletion of STING. And the knockout itself also led to an upregulation of the interferon-stimulated genes depicted in those 9 graphs on the left. And this interferon signature could also be completely reverted by the genetic codeletion of STING, but not by the genetic codeletion of either MUFS or MUT88 in gray. So for the conclusions, AD2 as an hitherto unappreciated DNase function, it is essential for the regulation of the nucleic acid sensing in monocytic cells with a specificity for the SIGA-STING pathway, and the auto-degradation of double-stranded DNA is the primary driver of the interferonopathy observed in data to patients. As further directions, we hope our data provides an important mechanistic insight into the pathogenesis of AD2 deficiency and other more common and complex autoinflammatory conditions, and opens the horizon for the development of personalized treatment approaches. I want to thank all the people involved in this project, especially Michele Proietti, Bodo Grimbacher, Max Warnke, and Ole Christian Greiner-Tollersrud.